So, as the CET examination is fast approaching and 80% of the questions for CET especially will be asked from the second PUC portions. So, I will complete the second PUC portions, then only I will take up the first PUC portions. Hitherto, I completed about 7 chapters in first PUC. So, as the CET fast approaching, let me continue with the second PUC's chapters. And I completed only 4 chapters in second PUC. Hence, I start with the 5th chapter, that is the principles of inheritance and variation. That is the subtitle under the genetics and evolution. So, branch of biology deals with the study of hereditary characters called genetics. Transmission of characters from parents to offspring called heredity. Differences brought to the offspring called variations. Variations are chiefly due to mutation. Mutation is sudden change in the hereditary material which is heritable. So hereditary materials are chromosomes. Chromosomes are called vehicles of heredity. Chromosomes made up of DNA and histones. So usually the students tell DNA and RNA. No, not correct. DNA and histones, chromosomes. Chromosomes are called vehicles of heredity. DNA is the hereditary material. And DNA made up of genes. Genes are called units of heredity. So sudden change in all these hereditary materials. That is the chromosomes, DNA and the genes, heritable, called mutation. So variations are chiefly occur because of mutation. One example given, that is Sahiwal. Sahiwal variations observed. So, individual with pair of alike genes called homozygous individuals. Individuals with pair of unlike genes on their chromosomes called heterozygous individuals. Offsprings obtained by a cross called hybrid. The character which will be expressed in hybrid called dominant character, the character which remains concealed in hybrid called recessive character. The external appearance of an individual called phenotype. Actual genetic composition of an individual is called genotype. These are some question and answers you should read in this lesson. So these are all the basics in this lesson. That is, once again I will repeat. Branch of biology deals with the study of hereditary characters called genetics. Transmission of characters or traits from parents to offspring called heredity. Differences brought to the offspring called variations. Variations are chiefly due to cross fertilization and mutation. Mutation is sudden change in hereditary material, that is chromosomes, DNA, genes, which are heritable, called mutation. Variations, for example, Sahiwal. Individuals with pair of alike genes called homozygous individuals, for example, capital letter TT, homozygous star, small letter TT, that is homozygous dwarf. And individuals with pair of unlike genes called heterozygous individuals, capital T and small t. Offspring obtained by a cross called hybrid. Character which is expressed in hybrid called dominant character. Character which remains concealed in hybrid called recessive character. So these are all the basic things, and the external appearance of an individual is called phenotype. Actual genetic composition is called genotype. External appearance of an individual, for example, in capital T and small t, because of law of dominance, they are usually tall. That is phenotype. But actual genetic composition is capital letter T and small letter t. Hence, they are called heterozygous individuals. Father of genetics, John Gregor Mendel. He conducted experiments on pea plants. He selected pea plants for his experiments. So, he conducted experiments for 7 years on pea plants, that is Pisum sativum of Fabaceae, and noticed 7 contrasting characters with reference to the size, or height of the plant, tall and dwarf. So towards this side, dominant character, towards this side, recessive character. And position of the flowers, axillary, terminal. Color of the flowers, purple, weight, and then the shape of the fruit. This is very, very important. Legume fruit or pod fruit. So dominant character is inflated character, and here it is constricted shape. Color of the fruit, green and yellow. Color of the seeds, yellow and green. And shape of the seeds, round and wrinkled. Shape of the fruit, if we ask, now prati sarti students round and wrinkled antare. Adukkoskara shape of the fruit very important, inflated and constricted. So conducted experiments for 7 years. Isvi kuda kottidane, onond sarti isvi kuda koಡ್ತಾನೆ. ಅದಕ್ಕೋಸ್ಕರ ಚೆನ್ನಾಗಿ ಜ್ಞಾಪಕ ಇಟ್ಕೋಬೇಕು. He conducted experiments for 7 years between 1856 to 1863. This is very important, already given in the NEET examination for 7 years ಅಂತ ಹೇಳಿ ಇಸ್ವಿ ಕೊಟ್ಟಿರುವಂತದ್ದು ಈಗಾಗಲೇ ಕೇಳಿ. ಈಸಿ ಜ್ಞಾಪಕ ಇಟ್ಕೊಳ್ಳೋಕೆ 1857 ಫಸ್ಟ್ ವಾರ್ ಆಫ್ ಇಂಡಿಪೆಂಡೆನ್ಸ್, 1856 ಸ್ಟಾರ್ಟ್ ಮಾಡ್ದ, ಅಲ್ಲಿಂದ 7 years 1863ಗೆ ಕಂಪ್ಲೀಟ್ ಆಯ್ತು. ಇವತ್ತಲ್ಲ ಇನ್ನೂ ಸ್ವಲ್ಪ ಚರಿತ್ರೆ ಸರಿಯಾಗಿ ಗೊತ್ತಿರೋದಾದ್ರೆ ನಿಮಗೆ 1863 ದಟ್ ಈಸ್ the year of birth of Swami Vivekananda, 1863. So hege 1856 to 1863. So next idella ಹೇಳಿದ್ರೆ, and then a cross involving single contrasting character called monohybrid cross and experiment called monohybrid experiment, and the ratio obtained from the monohybrid experiment is monohybrid ratio. So to begin with the first experiment, Wegener took a tall pea plant, homozygous tall pea plant, capital letter TT, and homozygous dwarf plant, small letter TT. Cross pollinated these two plants. Cross pollination, it involves, as you know very well, that is hybridization ಅಲ್ಲಿ ಹೇಳಿದೀನಿ ಈಗಾಗಲೇ. Cross pollination, it involves emasculation in one plant and then bagging with the butter paper of the gynoecium, and when the stigma attains the receptivity and pollen, when the pollen will be released, pollen gets matured and the stigma gets attained maturity. So removal of that, that is, next is dusting and rebagging. So after this, in the F1 generation, for his surprise, he found all tall plants, but they are heterozygous, capital T and small t. When the offsprings obtained in the plants obtained in the F1 generation, that is heterozygous individuals, are self-pollinated. In the F2 generation, results obtained in the ratio of 3:1, that is the phenotypic ratio 3 tall and 1 dwarf, and the genotypic ratio is 1:2:1, 1 homozygous tall, 2 heterozygous tall, and 1 homozygous dwarf. ಅದು ಈಕ್ವೇಷನ್ಸ್ ಎಲ್ಲ ಕೊಡ್ತಾನೆ, ಅದು ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್. ಸೊ ದಟ್ ಈಸ್ a plus b whole square is equals a square plus b square plus 2ab, ಅದರದೇ ಈಕ್ವೇಷನ್ ಕೊಟ್ಬಿಟ್ಟು half t ಅದನ್ನ ರೆಪ್ರೆಸೆಂಟ್ ಮಾಡಿರುವಂತ ತೋರಿಸ್ತೀನಿ, ಇಂಟು half small letter t square of this, that will give you quarter capital letter tt, half capital T and small t, quarter small letter tt. So this is about the monohybrid class, which is very important, and this equation also very important for the NEET examination. A class involving two contrasting characters called dihybrid class, and experiment called dihybrid experiment, and the ratio obtained from this is the dihybrid ratio. In this experiment, Wegener took a plant producing round and yellow seeds, round shaped, round shaped yellow colored ಅಂತ ಹೇಳೋದು ನೆಸಿಟಿ ಇಲ್ಲ, round yellow seeds ಅಂತ ಹೇಳ್ಬೋದು, and then wrinkled green seeds. So here it is represented by capital RR, capital YY, and small rr, small yy. So he cross-pollinated these two plants. So cross-pollination by the emasculation, and then after the emasculation, bagging, then dusting and rebagging, and then fruits obtained are sowing. Then in the F1 generation, all the plants produced round yellow seeds, but they are heterozygous. And in the F2 generation and in the F2 generation, when the plants obtained in the F1 generation are self, in the F2 generation, results obtained in the ratio of 9:3:3:1. 9:3:3:1. So this is the ratio obtained, that is phenotypic ratio. But there are 9 different varieties in genotypic ratios. That is also very important. How many varieties in the genotypic ratio of dihybrid experiment means? 9 different varieties. That is 1:2:2:4:2. 1:2:1:1. That is the genotypic ratio in the dihybrid experiment. After conducting these experiments, Gregor Mendel proposed 3 laws, that is the law of dominance. ಅದು ನಾನು ಒಂಥರ ಭರತನಾಟ್ಯ ಸ್ಟೈಲಲ್ಲಿ ಅದನ್ನು ಹೇಳಿದ್ರೆ ಎಲ್ಲ ಮಕ್ಕಳೆಲ್ಲ ಎಂಜಾಯ್ ಮಾಡಿದ್ದಾರೆ ಇದುವರೆಗೂ, ಅದಕ್ಕೋಸ್ಕರ ಅದನ್ನು ಹಾಗೆ ಮಾಡಿ. ನಾನು ಭರತನಾಟ್ಯ ಒಂದು 7 years ಕಲಿತೆ. ಯಾಕೆಂದರೆ ಅದೊಂದು ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಅಂತ, ಒಂದು ಎಕ್ಸರ್ಸೈಸ್ ಆಗುತ್ತೆ, ಬಾಡಿ ಸ್ವಲ್ಪ ಲೀನ್ ಆಗಿರೋದಕ್ಕೆ ಅಂತ ಪ್ರಯತ್ನ ಪಟ್ಟೆ. ಅದು ಆಮೇಲೆ ಬಿಟ್ಬಿಟ್ಟೆ, ಯಾಕೆಂದರೆ ನಡಿಗೆ ಚೇಂಜ್ ಆಗೋಯ್ತು, ಒಂದು ಲೇಡೀಸ್ ಥರ ನಡಿಗೆ ಬಂತು, ಅದಕ್ಕೋಸ್ಕರ ಅದನ್ನು ಬಿಟ್ಬಿಟ್ಟೆ. ಸೊ ಎನಿಹೌ ದ ಲಾ ಆಫ್ ಡಾಮಿನೆನ್ಸ್ of the 2 contrasting characters, ನೋಡಿ ಎಷ್ಟು ಚೆನ್ನಾಗಿರುತ್ತೆ, ಲಾ ಆಫ್ ಡಾಮಿನೆನ್ಸ್. Of the 2 contrasting characters, dominant will be expressed while the other remains concealed. ಇದನ್ನ ದಿದ್ದಿತ್ತೈ ಅಂತೀವಿ ಡ್ಯಾನ್ಸ್ ಅಲ್ಲಿ, ದಿದ್ದಿತ್ತೈ ಅಂತ. Of the 2 contrasting characters, dominant will be expressed while the other remains concealed. This is called law of dominance. And contrasting characters are separated out during the formation of gametes. This is called law of segregation of genes. It is also called law of purity of gametes. Separation and distribution of genes to the gametes is entirely independent of other genes. This is called law of independent of assortment. So, these 3 laws proposed by John Gregor Mendel. Once again, of the 2 contrasting characters, dominant will be expressed, the other remains concealed, called the law of dominance. Contrasting characters are separated out during the formation of gametes. This kind of separation and distribution of genes to the gametes is entirely independent of other genes. This is about the 3 laws proposed by John Gregor Mendel. So, till 1900, John Gregor Mendel's laws were followed. In 1900, Schamark of Austria, Carens of Germany, Hugo de Vries of Holland, they rediscovered the Mendelian laws. With the rediscovery of Mendelian laws, they came to know that laws of Gregor Mendel not universal. There are certain experiments which deviate from normal Mendelian laws. One such a deviation is incomplete dominance. So, according to law of dominance, of the 2 contrasting characters, dominant should be expressed, other remains concealed. But in incomplete dominance, dominant character will not be expressed completely. So, the dominant gene is masked by the recessive gene. As a result, in an incomplete dominance, intermediate forms appear. So, Carens did an experiment on the dog flower plant, Antirrhinum majus, or dog flower plant. So, in this experiment, in this incomplete dominance, dominant character will not be expressed completely and intermediate forms appear. So, in this experiment, that is in dog flower plant, also called snapdragon, technical name is Antirrhinum majus of Scrophulariaceae member, Antirrhinum majus. Snapdragon, dog flower plant. When a plant producing red color flower is made to cross with a plant producing white color flower. In the F1 generation, the plants should produce red color flowers according to law of dominance, but the plants produced the pink colored flowers. So this is because of incomplete dominance, where the dominant gene is masked by the recessive gene. As a result, intermediate forms appear, that is the plant produced during F1 generation, produce pink color flowers, intermediate forms. This is because of law of dominance. It is a deviation from normal Mendelian laws. And when the plants produced in the F1 generation are self-pollinated. In the F2 generation, results obtained in the ratio of 1:2:1. 1 with red color flowers, 2 with the heterozygous, that is the pink color flowers, and 1 with homozygous, white color flowers. That is, the 1:2:1 is the phenotypic ratio. And genotypic ratio is also same, 1:2:1. So, phenotypic ratio and genotypic ratios are identical, same in incomplete dominance. This is very, very important for the NEET examination. So, the class in which the phenotypic and genotypic ratios are identical, that is in an incomplete dominance, and even in test class also. So, if the hybrid obtained in the F1 generation, if the plant obtained in the F1 generation after classing the selected plants, and then if this hybrid is made to cross with homozygous recessive parent individual, this cross is called back cross or test cross. Here, the example given is a plant producing violet colored flower. So instead of capital letter VV, they used in the textbook, that is the capital letter WW for the plant producing violet colored flower, and the plant producing white colored flower, small letter WW, capital letter. And in the F1 generation, plants produced violet color flowers, that is capital W and small w heterozygous, according to law of dominance. If the hybrid obtained, that is in the F1 generation because of this class, made to cross with homozygous recessive parent individual, that is small letter WW, and in the F1 and the results obtained in the ratio of 1:1. So, this, because of test class, Gregor Mendel conducted a test class, so that is very important to know the correct genotype of the hybrids. If it is heterozygous, results obtained in the ratio of 1:1. Because capital letter W, small letter WW, and we can obtain the individuals in the same in the ratio of 1:1. If they are heterozygous individuals. If it is homozygous individuals, only one type homozygous individuals, only one type can be obtained. And so this is about the only similar type if it is heterozygous individuals. If the hybrid is the homozygous, it is only one type of results, and if it is heterozygote, the ratio obtained, results obtained is the ratio of 1:1. So to know the correct genotype of these individuals, Gregor Mendel conducted the test class. This is very, very important. And so in 1900, again, Karl Landsteiner discovered blood groups based on the type of sugar protrude out from the RBC. That is, the different blood groups in case of human beings, that is ABO, controlled by the two genes, genes I, capital letter I. Small letter I does not produce any the sugar. So, based on this, the blood groups are classified. If the A group will have capital letter I, that is, the genotype is IA, capital letter A, that is homozygous dominant. Or the A group may have IA, capital letter, small letter A, and capital letter A and small letter I. That is homozygous dominant or heterozygous dominant. Similarly, IB or IBI for B group. For AB group, it is capital letter IA, capital letter IB. They are called codominant. If the IA, IB together, they produce offsprings of their own kind. They express in their own kind. This is because of codominance, as there is no recessive individual. And the very important question for the NEET examination is, in blood groups, how many phenotypes and how many genotypes? The phenotypes are only 4. That is A group, B group, AB group, O group. That is the phenotype. Genotypic ratios are 6 because for A group, homozygous dominant, heterozygous dominant. For B group, homozygous dominant, heterozygous dominant. For AB group, codominant. For O group, it is homozygous recessive. Thus, there are 6 genotypes and 4 phenotypes in the blood groups. This is very important. And the inheritance. Fast I had there. A with A, AO. Now, and A with B, almost all types are produced if they are both are heterozygous individuals. And A with the AB, AB, A with O, AO, B with B, BO, B with AB, AB. Okay. So, B with B, BO. And then AB with AB, 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 AB with O. That is ARB. So, all the parents give on the blood groups and O with O. So, once again I will repeat. A with A is AO, A with B, A, B, AB, O, because if they are heterozygous individuals, A with AB, A, B, AB, A with O, AO, B with B, BO, B with AB, A, B, AB, and B with O, BO. And then AB with AB, A, B, R, AB, AB with O. Here it is ARB because when they come together, they express in their own way. This is because of codominance, and O with O is O. So, a pair of genes located on the homologous chromosomes which govern a particular character called alleles. 2 or more genes which govern a particular character called 2 or more genes, that is multiple alleles. And the phenomenon called multiple allelism or gene polymorphism or pleiotropy. Example, blood groups in the human beings, skin color in human beings, controlled by many genes, 2 or more genes. That is multiple allelism. 2 or more genes located on the homologous chromosomes which govern a particular character. So there are certain experiments which deviate from normal Mendelian laws. So another one important is the synthesis of starch in pea plant, pea seeds. That is another example where the one gene shows more than one effect. One gene showing more than one effect. That is, we can see in synthesis of pea seeds. This is also very important. And the plants having capital letter BB, that is, they are round and produce, they are more efficient. Starch grains are produced with large size starch grains, that is BB. And the small letter BB, wrinkled shape, but small letter starch grains are produced. So, before they attain the maturity, here somewhat not round in shape, but after attaining maturity, in case of capital letter BB, they attain the round shape with large size starch grains, and here after maturity, wrinkled only, but small sized, less efficient starch grains are produced in capital letter BB and small letter b. But the heterozygote produced shows both the dominant character and incomplete dominance. Dominance and incomplete dominance. Heterozygote obtained capital B, small b will have round shape, that is the dominant character, but the starch grains produced are intermediate size. This is incomplete dominance. Thus, dominance is not the autonomous feature of a single gene. Sometimes more than one phenotype influenced by the same gene. So these are all the other things. And what are all the things that I have left is how the dominant character, say for example, in the production of enzymes, So some alleles, they produce the enzyme as a whole, produce normal enzymes. They are dominant characters, or enzyme with less efficiency. And non-functional enzymes are produced because such allele definitely will have recessive character, or no enzyme is produced recessive character. So the inheritance is also depends upon these alleles. So multiple alleles of dominant type produce normal enzyme. So if the modified allele means non-functional enzyme and no enzyme at all. So these are all the things given in the textbook. This is only part A. Let me come to the next, that is chromosomal inheritance, in the part 2. So ಪ್ರತಿಯೊಂದು ಲೈನ್ ಓದಿ ಈಗ ನಿಮಗೆ CET ಹತ್ರ ಆಗ್ತಾ ಇರೋದ್ರಿಂದ ಸೆಕೆಂಡ್ ಇಯರ್ ಚಾಪ್ಟರ್ಸ್ ನಾನು ಪಾಠ ಮಾಡ್ತಾ ಹೋಗ್ತಾ ಇದ್ದೀನಿ. ಅದಕ್ಕೋಸ್ಕರ ದಯವಿಟ್ಟು ಅದನ್ನೆಲ್ಲನೂ ಚೆನ್ನಾಗಿ ಓದಿ. ಆಲ್ ದ ಬೆಸ್ಟ್ ಫಾರ್ ದ CET ಮತ್ತೆ ಪಾರ್ಟ್ 2 ಕಮೆಂಟ್ಸ್ ಮಾಡಿ.